This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only. Check out the new YouTube original channel UCTV Prime at youtube.com/UCTV Prime. Subscribe today to get new programs every week.
We all strive to live a long, prosperous, and healthy life. With advances in health and medical sciences, this goal is ever more attainable. The Sam and Rose Stein Institute for Research on Aging is a non-profit organized research unit under the auspices of the University of California at San Diego, committed to advancing lifelong health and independence through research, education, and patient care. To better empower and improve the lives of young and old alike, the Stein Institute presents the following program.
Without further ado, I'd like to introduce tonight's speaker. Our speaker is Dr. Cinnamon Bloss, who's going to talk to us about consumer genomics. What do people do with their genomes? Dr. Bloss got her bachelor's degree from Smith College in psychology. She received her PhD from the Joint Doctoral Program here at UCSD and San Diego State. She also completed her postdoctoral internship at the Scripps Research Institute in genomics and statistical genetics, so a very numbers-oriented person. And I just, with great pleasure that I introduce her to you, I have to put a caveat in that of all the many speakers I hear all the time, Dr. Bloss is phenomenal and great at taking very complex subject matter like statistical genetics and making it understandable for those of us who don't even know what that really means. So without further ado, I'd like to welcome Dr. Bloss.
It's a real pleasure to talk about genomics, a topic very near and dear, that's become very near and dear to my heart. So I hope that this will be an interesting foray for you all into this field as well. So when I give talks on genomics, and in particular consumer genomics, I always like to get some sense of what people know or have heard about consumer genomics, in particular because it's an area that's received a lot of attention in the lay media. So maybe I could just ask for a show of hands for anyone who's heard of what's called direct-to-consumer genomics. Have you heard of that term before? Okay, so a few people. All right. Well, excellent. It's good to have that going into the talk. So this just gives you a little bit of an overview of what I'd like to touch on today. First, just as to kind of get into things, touch on some of the historical points around genomic testing, or genetic testing really, then kind of get into direct-to-consumer genomics, and then sort of define it, in particular for those of you who haven't heard of it. And then to address some of the kind of the tricky and very controversial issues that have come up around consumer genomics. And then I'll touch on some other aspects of genomic testing as well as comment on where we are and where do we go from here. All right, so in this little cartoon that's depicted here, we see a person who goes to check their mail and they receive, their genome report, right? That says, they have, myopia, hoarding tendencies, absent-mindedness, and sort of a look of concern on their face, right? And so is this genetic risk testing of the future, or are we there now? Is this the genetic testing of the present? So with that backdrop, let's go back several years to the late 1800s when Mendel published the first theories of inheritance, so looking at his pea plants, and was really one of the first people to comment on inheritance and how it works. Later, theories of certain types of inheritance, modes of inheritance were published. And then taking us into kind of the early 1900s when the term gene was actually coined. And then a little bit later, the term genome was coined. to the 1950s, when the very famous paper came out by Watson and Crick with the help of Rosalind Franklin, who discovered sort of the structure of DNA, and this was just a landmark publication and continues to sort of inform the field of genomics even now. In terms of genetic testing, takes us to 1959 when Down syndrome was discovered to be a chromosomal abnormality. In 1960, the first prenatal sex determination was performed, and then amniocentesis was used for prenatal diagnosis of Downs. In the 1980s, the Huntington's disease gene was mapped to chromosome 4. This was actually the first, is considered to be sort of the first genetic test that was available, was a test for Huntington's disease. Then cystic fibrosis in the mid-80s was mapped to chromosome 7. Then going a little bit in a different direction, in the mid-1980s, Richard Buckland was the first person acquitted of a crime based on DNA, so a different type of genetic test. The first DNA microarray was put forth in the late 1980s. We'll talk a little bit more about what is a microarray. Finally, in 1990, the Human Genome Project was launched. In 1994, a very high penetrance gene was discovered that confers risk for breast cancer. What I just presented were sort of some kind of early concepts around genetics and genomics. In particular, sort of genetic tests for things where you can actually predict whether or not someone will develop a disease, like for instance with Huntington's disease. If a person has the Huntington's disease mutation, we know whether or not they will get the disease. But I think what's really important in understanding the genomic testing that's occurring now is this idea of, rather than sort of genomic predictive testing, the idea of genomic susceptibility testing, right? So you may have a genetic variant that confers some degree of susceptibility, but it's just that. It's a risk variant. It doesn't have sort of predictive validity, if you will. And so what's kind of brought us to that point? What's allowed us to determine variants that account for susceptibility to disease? Well, in large part, the Human Genome Project, which was completed, I mean, it's really still ongoing, but completed an initial draft of it in 2000. And then in 2004, something called the International HapMap Project was sort of ongoing, and they released not really a draft of the human genome, but more of a catalog of all the different types of variability in the genome. So the idea that I might have a certain risk variant at a particular position in the genome, whereas another person might have a different risk variant at that, or a different variant, excuse me, at that same position. And so it was this project that really kind of cataloged all of the variation that we now know exists in the human genome. And this was really critical for setting the stage for some of the genomic testing that we'll talk about tonight. And it also allowed what are known as genome-wide association studies. Well, basically, with the technological advances conferred by these two major initiatives, we're now able to, more rapidly and more cost-effectively than ever before, we're able to measure pretty much across a person's entire genome to assess all the different forms of variation they have at certain positions. And I stress that as well because it really is crucial to some of the concepts I'll be bringing up later. And so the first genome-wide association study was published in 2005, and actually this should be updated to 2011. Over 1,400 GWAS have been published identifying over 150 risk variants or susceptibility variants for more than 230 common diseases and traits, which is just an incredible advance in terms of our knowledge of the genome, in terms of our knowledge of human health. And so this is just a slide that depicts all of the different risk variants that have been identified across the genome. And let me just go through how you interpret this. So line here, it's a little bit hard to see, represents a chromosome, so chromosome 1. This is chromosome 2, 3, 4, 5, and then all the way down here to the X chromosome and Y chromosome. And then the little bubbles show where certain genetic variants have been identified that are known to associate or confer susceptibility to disease. And it just shows you that because we've been able to do these genome-wide scans, we've been able to identify susceptibility variants really across the entire genome. Okay, so I really want to draw out the distinction once again between the idea of more what we call Mendelian diseases versus complex diseases. And so complex diseases versus Mendelian are not inherited in predictable ways. And they're not caused by a single variant, or likely even a few variants. There's likely many variants that are responsible. So an example of a common complex disease would be Alzheimer's, or type 2 diabetes, for instance, or macular degeneration. These are all common complex diseases for which risk variants have been identified via GWAS. And complex diseases are characterized by having both genetic and environmental risk factors. This is important as well. So even if we did know, for instance, all of the genetic variants that were responsible for type 2 diabetes, we know that there are environmental risk factors as well. So even if you did know all of the genetic variants, you couldn't predict with absolute certainty whether or not someone would develop that condition. And as I mentioned, common complex diseases, have a genetic component that involves many variants, both common and rare. The field's sort of in its infancy, but we also believe that it's probably true that a number of genes interact to influence risk for common complex diseases as well. And we're really just at the beginning of being able to kind of sort that out, as well as gene-environment interactions. And this is really important. Even though, in my opinion, genome-wide association studies have been extraordinarily successful in identifying risk variants, the variants that have been identified only account for a very small fraction of the risk that's associated with these diseases. And this is important for when I'm going to start talking about genomic testing, because you could imagine that if you give somebody a susceptibility test, say for type 2 diabetes, and you're only able to sort of give them information about 2% of their genetic risk for type 2 diabetes, some people have argued, well, you're not giving them the full picture, the science isn't at a point where we can really sort of ethically give that information to people, it's not informative, and so on and so forth. So that's kind of an important, a really important distinction. And this is in contrast, again, to a condition like Huntington's disease, where if you have the mutation, you will get the condition, the disease. So it's important. And so some examples, as I mentioned, I also mentioned type 2 diabetes, macular degeneration, Alzheimer's disease. Here we have, and this is for the common late onset version of Alzheimer's. This isn't for early onset familial Alzheimer's. We estimate about a 38% of the disease is due to environmental factors versus about 62% is thought to be due to genetic factors. And this slide again just kind of underscores some of the points that I was making earlier. So we have, for Huntington's disease, really a predictive test. We know the mode of inheritance, we can measure the mutation, and we can give that information back to someone, and it will kind of be a sure thing, so to speak. There are other conditions where we know markers that have a very high penetrance, so they actually account for, not all of the disease, but a high proportion of it, like markers that are responsible for familial breast and ovarian cancer, like the BRCA markers, that confer about a 50 to 80% chance of, for instance, breast cancer. And again, this is kind of contrasted to common complex diseases, where, as I mentioned, the known genetic markers account for less than 5 to 10% of the genetic risk of the disease. Some studies have also suggested that the genetic variants that have been identified for these conditions is actually redundant with a family history. So, for instance, if you know your family history for macular degeneration, then you kind of already know everything that the risk variants would be able to tell you anyways. So people have argued that, well, we shouldn't give this information back to people because it's redundant with family history. Kind of ending here in my description of common complex diseases, testing for these for genetic risk for these diseases is very controversial at present, but it is being performed in some contexts, and it's exactly that type of testing that I'm going to be talking about here tonight. Now I'd like to move into talking a little bit about genomic testing. I hope that I've given you some idea about why it's unique relative to single gene testing. So with the genome, we're talking about all of the genes in the genome. With single gene diseases, we're talking about one risk variant. So I'll switch now and talk a little bit about genomic testing. And again, this is incredibly controversial. It's a very controversial field. But basically, what's happening now is that research findings from genome-wide association studies are now being sort of used by companies, so industry, to offer genomic testing for complex diseases, all right? And there's just really a lot of debate regarding this. In fact, if you search on direct-to-consumer on the internet, it's incredible the number of hits that you get and just the range of commentaries and so on. I would encourage you to do that if you're interested. Let's go through sort of the pros and cons, I would say. So people who oppose genomic testing in this context, in the idea that you can get this testing from a company, one major criticism is the way that it's delivered. So these companies are doing what's called DTC, or direct-to-consumer. So I'll go through this, but basically you can go on the internet, you can find one of these companies, you can pay your money with your credit card, they'll send you what's called a spit kit, right? And you get it in the mail. So in the comfort of your own home, you can move your cheeks and generate your saliva and spit in the tube, send it back to them, and 4 to 6 weeks later you'll get an email with your genomic risk report in it. Very controversial. So in that whole process that I just described, you never heard me say a physician was involved, right? Or a genetic counselor was involved. And this is a huge, there are many criticisms of this type of testing, but this is one big one, that it's direct to consumer and that doesn't involve a healthcare provider. Other people argue that it lacks demonstrated validity, the kind of validity and utility that we would require of a medical test, if you consider this to be a medical test. That it's expensive, that perhaps this contributes to a divide in terms of some people who can afford the testing are able to get it, but others who can't don't have access. The way in which the results are communicated, some people have argued that it's just a very difficult topic to try to explain to somebody, and therefore, this is sort of another argument for why a physician should be involved, or why a genetic counselor should be involved. And then finally, another major criticism is that this maybe has the potential to cause harm, right? So if somebody, for instance, gets a risk result back that says you're at high risk for type 2 diabetes or for Alzheimer's, for instance, that this has the potential to cause a lot of anxiety, right? So that's another concern. The people who are in favor of sort of continued direct-to-consumer testing have a number of arguments as well. They argue that, well, if you empower consumers with their own health information, with their own genome, that you can actually empower them to be better advocates for their own health care, right? So one issue is that, sort of related to this, is that physicians as a group, they've been surveyed, and it's been found that that actually 90% of physicians say that they don't feel like they understand enough about genomics to be able to kind of use it with their patients, right? So some people have argued that by giving the consumer direct access to this, they can just be better advocates for their own health. Other people have argued that that it gives somebody the opportunity or perhaps the motivation to make more healthful lifestyle choices. So if I find out I'm at increased risk for type 2 diabetes, maybe I would be more likely to watch my diet or more likely to exercise more. And then finally, another argument in favor is just that, kind of this democratic argument that people have a right to their own information. And who is anyone, physicians, genetic counselors, the government, policymakers, whomever, what is it their right to keep a person from accessing information from their own DNA? So, so those are some of the arguments for and against. So there's two kind of main companies in this space, well, actually 3 main companies in this space right now. One's called 23andMe, 23 pairs of chromosomes, that's where they get the name. Another is called Navigenics, and then finally another company is called Pathway Genomics. They're actually based right here in San Diego. And this is an ad taken from the Navigenics website. They say there's DNA and then there's what you do with it. So their idea is really, centered around that, people can use their DNA to improve their own health. And you see, the woman marching up the double helix ladder, getting lots of exercise, and so on and so forth. So as I mentioned, this type of testing, it kind of came on the market right around 2007, so it's still a relatively recent occurrence. But it was a number of years ago touted as one of the best inventions of the year by Time magazine. So just to kind of underscore the attention that it's received in the media. But I've alluded to the intense controversy over this testing. And here I show just a handful of the commentaries and opinion pieces that have come out about genomic testing. So, people calling for the regulation of direct-to-consumer genetic testing. Are genetic tests predictive or premature? Question mark. Rational responses to the gathering storm of personal genomics. Personal genomics information can be harmful. And so forth. So, and these are all, sort of prominent thinkers in bioethics, in genomics, in, public health. So this has really generated a lot of debate among many groups of people. So, and if you didn't think that the controversy among scientists and clinicians was enough, the controversy, and some of you may remember this from the news, it received a fair amount of news, science news anyways. The controversy sort of intensified in 2010 when Pathway Genomics, one of the companies that I mentioned, they announced a partnership with Walgreens to actually sell their genetic testing kit at over 6,000 of their retail stores. And this would have, so this received coverage in the New York Times. This would have marked the first time, really, that genomic testing moved from sort of the internet commerce space to, retail on the shelves at retail stores. And, I think the FDA and other perhaps regulatory agencies had been kind of keeping their eye on direct-to-consumer testing for a while, unsure perhaps of how to go forward, how to regulate. But when this partnership was announced, it really started kind of a whole flurry of activity in this space. And I think they were in a position where they couldn't really ignore it anymore, sort of. So the FDA notified Pathway that their test kit appeared to meet criteria for medical device and required FDA approval, which it did not have previously. The FDA then went ahead in June of 2010, sent a similar letter to other direct-to-consumer companies, sort of indicating that they needed to obtain FDA approval, which, again, they had not previously. And then finally, in July, there were public meetings that the FDA held to gather information about how these tests should be regulated. And then finally, in late July, there was a congressional committee hearing on this, and they actually unveiled the results of sort of a federal sting operation where they had people who were working for the General Accounting Office actually go undercover to the companies and basically found some evidence of deceptive marketing practices and so forth. I mean, that report was criticized quite a bit. I think it was a very small sample. I don't want to stand up here and give you the impression that these companies are sort of deceptive or anything like that. I think they are all, I mean, not all of them, but the main ones are trying to do good science and are trying to provide, I mean, they're a business, so they're trying to make money, but I think they also are trying to sell the best product that they can. But nevertheless, this was a huge low moment, let's say, for direct-to-consumer testing. And the FDA has some ongoing things that they're trying to do to figure out how best to regulate. I think one thing that could be on the horizon is that they won't necessarily demand that companies shut down their testing operation, but that they may start to require the involvement of a physician. So that might be one approach that they would take. Other companies have actually changed their business model. So Pathway Genomics was the company I mentioned here in San Diego. And they were direct-to-consumer prior to all of this, but have changed their business model to actually, now you actually have to go through it. You do have to go through a physician. So you're raising a recent book by Eric Topol, The Creative Destruction of Medicine, where he talks about having his own genome measured. And the question was, what's the cost associated with doing that? So I don't want to get too far off topic, but one distinction I will make, because that's a great question, is there's sort of different ways of measuring the genome. You can get sort of what's called a SNP profile, which is what most of these companies give you. But you can also get every single base of your genome measured, which is called whole genome sequencing. And so I actually work with Dr. Topol, and I know that he's had SNP scans from all of the major companies, and to my knowledge, he has not had his whole genome sequenced, but I could be wrong about that. But anyways, the SNP scans, which is what I'm talking about up here, they range a little bit. 23andMe, I think, basically offers their service for about $99 for their SNP scan. You can get a whole genome sequence now. That's more expensive though, and to my knowledge, I don't know if there's direct-to-consumer whole genome sequencing, but that's about $4,000 or $5,000. The cost is declining rapidly. But if you want to go through one of these companies, it ranges. It's right around $100, really. Okay, so I just want to make this a little less abstract, and so I'm going to take you through, how it happens. All right, so this is taken from the website of 23andMe, where here you can sort of see my name up here because I've had my own genome tested through 23andMe. And basically, as I said, you order your kit, you pay your money, you get your kit in the mail, you spit into the tube, make sure you don't eat anything ahead of time or brush your teeth, and then you send your kit back. They analyze it, and then you get an email, and you can click on the link that's in the email, and it will show you your risks for diseases and a number of other things, which I'll show you in a moment. So here is a screenshot of the email I received when I did this, which was back in June, actually. So, dear Cinnamon Bloss, data is now available for you. It's available in your online account, blah, blah, blah. Just click here and you can access it. So I did, and this is my part of my health overview from 23andMe, and it's a little bit hard to see over here, but 23andMe really provides you with a lot of different things. They're probably the most comprehensive direct-to-consumer company that you can go through right now, just in terms of the different sort of traits and diseases and drug response items that you can get information for. They also give you information on your genetic ancestry. So through your genome, you can actually make some determinations around where most of the chunks of your genome kind of originated from in terms of worldwide locations. So I'll show you some of my results for that as well. But I'm not sure how readable this is for all of you, but not at all. Okay, sorry about that. Basically, here are my disease risks. I'm at elevated risk for coronary heart disease. But what I'll point out here is that it says that my risk is 38%, but the average risk is 24%. So the average person walking the planet has a certain amount of risk for coronary disease as well, right? And I happen to have some SNP variants that put me at increased risk, but the increase is 12% based on their algorithm, and then everything else is much lower than that. So what are these numbers in parentheses? And that's the total number of diseases that they test for, so 116 diseases. And in fact, that number goes up all the time because the scientific literature is expanding all the time. And so 23andMe is very good about bringing in new scientific findings into their testing protocol. And you can click on, each of these is actually a clickable link, and so if you click on the link, it actually tells you more about, like, well, what is coronary disease? What are the genetic risk variants that have been identified? It's actually quite educational. So the company provides you with updates, and so not only do you get that initial email that I mentioned, but you also get periodic emails, and they say something like, 6 new discoveries have been made about your DNA. And so you can click on that. You might be at work when you get the email. You can decide whether you want to open it then or wait. But I think the point that you're making is that they are, at this point, charging for that kind of ongoing subscription service. So, yeah. This is disease risks. This is traits, so things like hair color and eye color. They give you, what's your likely predicted eye color and other sorts of things. This is carrier status, and so it basically gives you information about whether or not you carry risk variants that are thought to be kind of more highly penetrant for certain conditions, so that have more kind of predictive power. And then the other one down here is for drug response, and I'll talk about this a little bit later if I have time. But I think that this drug response area is actually one of the areas where genomic testing is sort of poised to actually could be very beneficial for people. Even now, I think that it's my opinion that the science is there to really to impact healthcare. And basically, what this test tells you is, we know certain genetic variants are associated with how people respond to certain medications, like clopidogrel or with Plavix, or Coumadin, warfarin, a blood thinner. And I think having genetic information for something like Coumadin can allow a patient and their physician to dose the drug much more accurately up front, and also to avoid side effects associated with certain drugs. The question is well, what about genetic ancestry? And I have a slide a little bit later that I'll show on that. But basically, because of there are methods now that allow scientists to basically determine where kind of major chunks of your genome originated. Right? So are your ancestors mostly from Northern Europe, or are they mostly from Africa? And we can actually make those determinations by analyzing the genome. And so what 23andMe does, which is actually very interesting, is they do this analysis as well, in addition to the disease risk analysis. And so they'll tell you, based on your genome, where your ancestors are mostly predicted to be from. All right, so I'm going to maybe go through some things kind of quickly. The reason I'm kind of going into all of this is because at Scripps, which is where where my lab is located, we've actually pursued some studies to start to sort of inform how we should deal with direct-to-consumer testing. So, all of the arguments that I mentioned, kind of for and against, those are all potential research questions where we need data to kind of inform whether or not that's the case or not. So for instance, if we think that this type of testing is going to cause anxiety in people, or if we think it is going to motivate healthful behavior changes, well, we need, instead of just kind of theorizing about that, we need to try to obtain data and see if that's the case. So with the Scripps Genomic Health Initiative, we tried to do that. It's a longitudinal study of people who underwent testing with one of the company's tests, the Navigenics Health Compass. And we basically looked at outcomes in 3 areas. Did the testing result in increased anxiety? Did it result in any changes to diet and exercise? And then finally, another area where you could imagine there would be a behavioral change around genomic testing is around clinical screening test use, right? So if I were to learn that I had an increased risk for breast cancer, let's say, maybe I would be more likely to want to get mammograms more frequently. And so another criticism of the testing is that, well, it's going to cause people to kind of raid the medical commons, right, and increase healthcare costs because people are going to be concerned and want increased screening. So that was part of our study as well. And so this just kind of outlines what we did. We had people complete a baseline health assessment. They received their personal genomic test, and then we did 2 follow-ups at 3 months and at 12 months. The question was, did they pay for it? And we wanted the study to have sort of some ecological validity. They didn't pay the current commercial value that was being charged for the test, but they did have a copay, and that copay ranged at the time. So this was going on in 2008. At that time, genomic testing was much more expensive than it is now, and the cost ranged from about $150 to about $400. Yeah. And these are just the basic areas where we collected outcome data. So, I showed my test report with 23andMe. This is just an example test report from Navigenics. The format is different, but the content is very similar. So they give you, here's your results for type 2 diabetes. You're at this percentage, 45%, versus the average is at 30%. It's the same idea. And, you can also see that, they have different risk formats. And one of the main questions about this testing is, well, how do you communicate these findings? That's an open question in the field right now. And I don't think anyone knows what the best answer is, but it's been kind of discussed nonetheless. And this study also had a sort of a genetic counseling component as well. So one issue with our study, we had about 2,000 people, over 2,000 people that we studied in this context. And we had quite an age range, 19 to 85. But one issue has been that the study sample was quite wealthy and also very highly educated. So those are sort of some caveats to keep in mind when I show you the results. One limitation might be that, well, you would see different results if you had a different study population. So, okay, so here, the question is, what happens when someone gets their genome, right? So, here's somebody's double helix DNA, and they're saying, you're at risk for something, lay off the burgers, right? Change your diet, become more healthy. Well, do people actually really do that is the question. And so we published these results last year, so they're available if anyone's, I don't know if anyone's interested in a scientific article, but I'd be happy to share that with the group. And this basically shows what were our outcomes in our main areas of interest. And so really, we didn't see any changes in anxiety, any changes in diet, any changes in exercise. But what we did see was people reported that they intended to get screening tests with greater frequency. They weren't actually doing it, but they said, yeah, I got my genetic risk result, and I think I'm going to be more compliant with getting my colonoscopy. That was kind of our main finding. And so just to kind of underscore that, especially since the psychological component has been a big issue in the field, really here's kind of the flat line that we saw. Here's anxiety on the y-axis and months in the x-axis. And really just no changes at all. Same is true with diet, which is disappointing for those who thought that genetic testing would be highly motivating, but it really wasn't, which was unfortunate. But and then finally, this is kind of a busy table, but basically the bottom line here is that, for several, we assessed several screening tests. We matched the screening test with the condition which would be sort of appropriate for that screening test, and then asked this question, well, do you intend to get that screening test with greater frequency? And so, for instance, here's the colon cancer and colonoscopy relationship that I brought up earlier. This is basically showing that for people who received an increased risk for colonoscopy, they were more likely to say, yeah, I'm going to get my colonoscopy with greater frequency. Okay, and then I wanted to, these data have not been published, but I did want to present them, given that we're here at Stein. So, you could imagine that genomic information might be really different, have different meaning to a 19-year-old versus an 85-year-old, right? So one issue is that, that's been talked about in the literature that I've thought quite a lot about is that, by the time a person reaches age 80, let's say, he or she may already know a lot of, what diseases that they're going to develop. And so, maybe there are fewer surprises versus a 20-year-old who may, be concerned about the future and not know sort of what it holds. So we've done some analyses by age, but haven't really seen, we've seen some effects, but here we've looked at sort of anxiety before and after testing. So the green is before testing, the yellow is after, and there's no difference across the age span. They're all pretty much level. But here we have sort of another measure of anxiety, which is called test-related distress. Here we're actually seeing that for people who are at increased risk for Alzheimer's disease, that they actually did show a difference in how distressing the test was as a function of age. And so what we saw was that people who are like between 40 and 60, those people who received increased risk for Alzheimer's, the test was a little bit more distressing to them than people in other age groups. So we also asked about sort of the natural sharing so this is direct-to-consumer testing, right? A physician is not involved. But you could imagine that for some people, they're naturally going to take their genomic test report to their physician. And in fact, that's been another criticism of direct-to-consumer testing, because the criticism is that, well, we're going to get a ton of people who are going to take this report that's like, 60 pages long to their physician, and, what was supposed to be, the 7-minute visit that you get at your physician's office is going to, the physicians are then going to have to spend a lot more time with patients. So we did kind of assess this as well. About 26% of our samples shared with their personal physician. And we actually saw some slight differences where people who shared with their physician were actually more likely to make some positive behavioral changes. So, that's a finding that we're kind of following up on more currently. Okay. So I know that's a lot of information, and I skipped over a few things. Before ending, I just want to comment on, well, where do we go from here? First, I'll just say that our study was really the first study to look at response to consumer genomic testing, and I think we did provide some important data on some of the issues, but there needs to be a lot more work in this area. So what are some future directions? Well, what about this issue of using genomic information for primary prevention? People have said, as I mentioned, that, well, genomics will motivate people to have a better diet or a better exercise routine, but we really didn't find any evidence of that in our study. And so one issue that has kind of been of interest to me is the idea that maybe there are critical periods in the lifespan where people are sort of more malleable in terms of adopting healthy behaviors. And we know this to be true, that younger people, that's the time in life when we sort of set our habits and so on. And so, but one issue is that, well, we could ask questions about giving genomic information to younger people or even to minors, but if you think that direct-to-consumer testing is controversial in adults, like, it's even much more controversial in children. So But nevertheless, and another study recently came out where they surveyed parents and found that actually parents do want genomic tests to be available for their children. So it's, again, a complicated area. But one way that we tried to explore this is we've started a study with high school students trying to gauge what their thoughts are, what their views are on genomics. And this has been done at the high-tech high charter schools down in Point Loma. We've asked them questions to just kind of gauge their knowledge. Many health problems are caused by a combination of genes, the environment, and lifestyle. All of them said true, the majority of them said true. If you have a gene that increases your risk of skin cancer, you can't do anything to prevent getting skin cancer. The majority of them correctly answered false. So kind of somewhat providing evidence against the idea that teenagers may not be able to understand the information. One thing we did was showed them an example drug response report, and these data are still being analyzed, but I'll share some quotes with you. Here's one: knowing this information early on, you're very well prepared later in life to know how to take care of your health. I think it should be available. I think someone my age would have the ability not to overreact, and if they don't understand it, they would know how to get further information. And then another quote as well. Another area that I think has been kind of interest to me as well is, genomic information is inherently shared, right? We share our genomic information with our family, with our biological family, that is. And so when you consider that, if one person in a family elects to be tested, how does this impact other family members, right? Because if I'm tested and my husband's tested, then we're going to know, a fair amount about, any offspring that we may have, any children. And I have a kind of a story about this. This is my carrier status testing from 23andMe, where I have been identified to carry a variant for what's called hemochromatosis. And so this is a condition where if my husband also carries a variant, that our children would have a significant risk of inheriting or developing this condition. And we welcomed our son a year ago. It's my husband and my son. And I got this in my email, right? I got my email and I see that I carry this variant. So, I immediately send him an email and I'm like, do you carry the hemochromatosis variant? And, luckily he didn't. But it was kind of an interesting exchange. So I think another area where we'll need to consider for genomics in the future. So just to conclude, I think in our study we found minimal impacts in our areas that we looked at in adults of genomic testing, which based on our data is consistent with continuing direct access of genomic information to consumers. So our data support the idea that consumers should be able to access this information directly without the involvement of a physician. But I don't know the extent to which that's going to happen. I actually testified before one panel of the FDA back in March of this year about our study. But as I said, we're still waiting to hear exactly how they're going to be handling this information or this type of testing. But with that said, I think, again, there are many, many questions that remain to be answered, and we need a lot more kind of research in this area. We may want to consider minors, parents, families. What are the issues there? How can we educate physicians to kind of use this information? And then also anticipate issues, because the technology is still rapidly improving. And although we can get these SNP scans through direct-to-consumer companies, the volume of genomic information that we're going to be able to measure is increasing all the time. And so, we need to consider the availability of genomic information with even better technologies in the future. And then also policymaking and regulation issues, and these are just some of the sort of legislative acts and initiatives that have already been initiated around genomics. And I'd just like to acknowledge my collaborators, somebody asked me about Eric Topol. I've worked very closely with him on this project, and a number of other people as well, and also our funding sources from the NIH and Scripps. The question, I just want to repeat the question, is around sort of the reliability of the tests, also issues around taking into account other factors like epigenetics. And so you're pinpointing another criticism of this testing that I didn't explicitly mention, but the fact that epigenetic factors contribute to the risk for these diseases as well, and these tests don't at present account for those. So I showed those pie charts where we know that, for instance, like with type 2 diabetes, there's some part of the disease that's environmental and there's some part that's genetic. And so when people talk about motivating behavior, that genomics might be able to motivate behavior change, what they mean is that if somebody learns they're at increased genetic risk, well, maybe they can decrease their risk on the environmental side by changing their diet. And so the expectation that has not been, that I'm aware of, that the changing the diet would result in other alterations in epigenetic factors, but that's certainly something that I'm quite certain will come into play in a few years. So it's a little bit confusing because both are often referred to as genome-wide tests, but the difference is it's somewhat subtle, but basically one is a SNP test, so SNP, SNP, yeah, and it stands for single nucleotide polymorphism. And it's not critical that SNP is fine, that's what everybody says, but basically the tests that I predominantly focused on here are SNP scans. So they measure SNPs across the whole genome, versus whole genome sequencing measures all the types of variation across the genome. And SNP scans are much cheaper right now relative to whole genome sequencing. I think what you're asking about is family history of disease, right? I know you use the word ancestry, but you're not talking about where people are from. You're talking about the disease risk in families, right?
I don't know where they came from. Yeah.
Okay. So I think one major criticism of these tests is that people do already know their family history. They do already know if their parent or their grandparent had a certain disease like macular degeneration or like Alzheimer's. And so a big question is, well, what does this really add beyond that? And I think that's kind of the question that you're asking right now. And I can tell you that there have been some studies that have tried to statistically parse that out, to say, well, is there a part of it that could be newly discovered via SNPs, via genome scans, in addition to what you already know about family history. And I'm sorry to report that for the most part, those studies have determined that family history is pretty much, based on the variants that we know now, are highly redundant with SNP scans. So that's a long way of answering your question, but at this point in time, I think for disease risk, there isn't necessarily a lot of additional benefit, right? But what I will say, you didn't ask me about this, but I will say that I still think there's a lot of value in getting this type of information, especially for drug response. So, drug response isn't necessarily the kind of thing that you track in the context of a family history. I mean, for instance, I have no idea how somebody in my family responded to Coumadin or clopidogrel. But it doesn't change the fact that someday I might need that drug. And I think that genetic information can be very helpful and very powerful in those instances.
But your statistic there indicated that 67% of the risk factors for Alzheimer's comes from our genes.
Mm-hmm.
So from our genetic background.
Mm-hmm.
So now you say that they found out this does not mean very much.
Well, it's a tough thing to kind of explain, but the issue is that we know what proportion of a disease is genetic from studies like twin studies and family studies. And we're able to kind of determine that when you study twins, what proportion of a disease is genetic, right? But that doesn't mean that we know the specific genes that are responsible. We just know there's some proportion that's genetic. So what I'm saying is that that 64% of Alzheimer's that's genetic, we only have identified the specific genes involved for a small proportion of that. Does that make sense? So there remains a number of variants that are yet to be discovered. And again, that's a criticism of these tests. Well, we don't have all the information, so maybe we're giving people results prematurely. I think maybe another question you're getting at, which is another good one, and you can correct me if I'm wrong, is the actionability of the diseases for which you're being tested, right? So Alzheimer's, some people argue that there are things you can do to sort of mitigate your risk, sort of exercise your brain. But I think what you're saying is that there are no treatments, right? And so what's the benefit in the case where you have a disease, you learn your risk for that disease, but there's no actions you can take? And so that's been another criticism. And I think Alzheimer's is one unfortunate example of a complex disease where there is not yet a lot that can be done because we don't have good treatments. But there are other conditions, complex conditions, where there are a lot of actions you can take. And so, things like type 2 diabetes, I mean, studies have shown exercise, diet, those sorts of things can mitigate risk. And so, I think that's one message I'd like to impart. I hope that answered your question. Your actual genetic sequence doesn't change. So, yeah, yeah. There's other sort of like the other omics that do kind of change over time. Things like epigenomics. But your actual sequence, which is what's being tested here, it remains the same. So the question was, are they adding new conditions? And the answer is yes, they are. And basically, the adding of new conditions is directly parallels the scientific literature. So as new discoveries are made in the scientific literature, 23andMe, for instance, will incorporate those variants into their test. Thank you. Thank you.